regard to this particular pest and the various strategies for management of this pest. And I hope and I understand that based on this particular questionnaire, Dr. Padmakumari will be making a short presentation. We have amongst us eminent personalities, Dr. J.S. Bentur, who headed entomology the department here, and then also coordinated research in entomology, besides working as principal scientist. And sir has also been my teacher with regard to Gall Midge. And Dr. Bentur has spent 3 decades studying Rice Gall Midge interactions. And we also have amongst us another panelist, Dr. Suresh Nair, who is currently Arturo Falaschi Emeritus Scientist from ICGB. And we are thankful to Dr. Arvind Kumar, Deputy Director General ICAR IIRR, who started his career with respect to research on Rice Gall Midge at IGKV Raipur. Sir, we are very thankful to you for joining us for this important brainstorming session and then sharing your viewpoints despite your busy schedule. And we also have amongst us Dr. C.H.V. Durgarani Madam, who is Professor and Head Biotechnology PGTSAU. Madam has done significant work related to molecular breeding of Gall Midge. And we also have amongst us Dr. Vikas Kumar Singh, who is IRRI South Asia Lead at ICAR Campus, Hyderabad. And I understand that IRRI is focusing on a major program on direct seeded rice. And I also understand that Gall Midge is expected to be one of the major or leading pests in many upland conditions wherein direct seeded rice is being practiced. And I'm also thankful to Dr. Shyam Kumar, an eminent expert breeder on Rice Gall Midge, who is presently Associate Director of Research, ZARS in Maharashtra. Along with all these panelists, we also have a galaxy of scientists who are basically entomologists and then breeders, directly or indirectly associated with research on Gall Midge. And I'm sure that their expertise and knowledge would help in addressing the issue of timely and critical management of this. So far, we consider this as early stage pest through fruitful discussion. And we are sitting here because from about 15 of our research stations encompassing 12 states, we are receiving reports of resurgence of this particular pest in the last, say, 5 to 7 years. And what we are also seeing is there's a change in the time and intensity of the incidence. In many traditional endemic areas, the pest, in addition to affecting rice crop in the early stage, it is also continuing beyond vegetative stage. And there are reports wherein we are hearing incidents even at reproductive stage also. And reports of major incidents have been received from large tracts of Tungabhadra Command area, Kaveri Basin, and also several regions of Telangana and other states. And fortunately, this is one of the pests, thanks to intensive efforts carried out by Dr. Bentu, Dr. Suresh Nair and team. We have got extensive knowledge on the mechanisms of host plant resistance and also about the genes and then the genetic sources conferring resistance. The service, there is some understanding with respect to insect virulence also. Rice breeding lines with Gall Midge resistance, as mentioned by Dr. Jansi Lakshmi, we have got in many elite genetic backgrounds which are ready for deployment. With the proposed or ban and also phasing out of some of the granular insecticides like carbofuran, the chance of deployment of insecticide by farmers is also very limited. Understand that in this context, we are gathered here today for this brainstorming session to have a relook at the available options and also to discuss this particular issue. I now invite Dr. A.P. Padmakumari to make a brief presentation on this particular issue based on information that has been gathered so far. Thank you, madam.
Thank you, sir. Thank you very much. Respected director, dignitaries in the online mode, my senior colleagues, panelists, my dear cooperators, fellow scientists. My
Anyone confirm whether the screen is visible? Can anyone, any online? Okay, okay.
It is visible.
Yeah. Thank you, ma'am. We can go ahead.
Yes, screen is visible.
Thank you, sir. A very good morning to you all and a warm welcome to this brainstorming session. As detailed by our director, we are having a relook at the status of rice gall midge as a pest and what we should do to tackle this. Today we are here to discuss the various options. We formulated a questionnaire and circulated among all our entomology cooperators, and we have got some answers. So I have summarized them and I'll be putting forth to you all the various options that are available, their thoughts on this particular pest, along with the knowledge that has been generated over a decade. So I'll just summarize in 20 slides what is the exact status to keep the ball rolling for the further discussions. Next slide, please. So Asian rice gall midge, actually among the 29 species of Ortsolia, we see that only 2 species are pest on rice. Out of that, Asian rice gall midge, that is Ortsolia oryzae, is confined to, is present in India and Southeast Asian countries. And it has been predicted, the losses have due to insect pests have been estimated at 25%. But losses due to gall midge, out of this 25% is estimated at 15 to 60% way back in 2003. And in 2010, Dhaliwal and co-workers predicted that there will be an increase in gall midge incidence with the climate change. And if you look at the pest-rice interaction, gall midge is a pest rise from nursery to tillering stage, which causes significant losses. After tillering stage also, it survives at a reproductive phase, but we do not perceive it as a loss, but the insect will be able to survive and complete its life cycle. And from the data that is, from the ICAR data from 1967 to 1992, it was calculated that the loss would result in a grain yield, and it was much more in the rain-fed ecosystems, leading to 240 kg per hectare, with 1% silver shoot damage, unlike an irrigated ecosystem where it is 147 kg per hectare. And the pest was reported to cause more damage on rain-fed crop than on irrigated crop. And infestation in nursery may lead to improper crop establishment in the main field, where the damage goes unnoticed, and sometimes it leads to ploughing back of the field also, which has not yet been quantified how many fields have been ploughed back. Next slide, please. And if we look at this is all old information, but for the newcomers, I'm just briefing it. The pointer is that. Mouse. So if you look at the microphone. Here, if we look at the microphone. Here, if you look at the biology of the pest, it is the eggs that are laid on the leaf, and the moisture or the humidity is the crucial factor for the movement of the maggot. And if here it is very crucial, if there is no movement of the maggot into the tillers, into the base of the tiller, there the establishment of the maggot would fail. And this is the maggot that feeds on the growing meristematic tissue. And after feeding for 12 to 15 days, it pupates. This pupa is very active and it wriggles through the gall to the tip, and there it makes an incision and emerges out. So this is a very active pupa, and it can move throughout the gall. And there is sexual dimorphism. The adults are bigger and the male is the smaller one. And the length of the silver shoot, the color of the silver shoot, and the size all depend on the variety that has been infested. And you can see the extent of gall midge weather. This is a photo from Gangavati where there was a very severe incidence in 2020. And here the gall formation requires a highly nutritious plant because it has to develop into a gall and the insect has to survive and feed. So if the plants are very healthy, then there will be more gall development, and each gall will have one insect. Though at the time of egg laying, there will be many maggots that will be reaching the meristem. There will be one insect per tiller. And we have observed up to 1 to 14 galls per hill in endemic areas during our surveys and our monitoring tools. And if you look at the infestation of Gall Midge, we can see that all the coastal areas are infected. And from our survey with our cooperators, we see that not much insect is present, but it is not alarming in the northeast. It is very alarming in the command areas of Tungabhadra, Kaveri, and Andhra Pradesh and Telangana coastal areas, and Karnataka also. But otherwise, in Kerala and Kerala, resistant varieties are grown, so it is not a big problem, though the insect incidence is evident. Now, coming to the factors favoring Gall Midge incidence, where during the survey, all these points have emerged. Knowledge on these factors is already available, that is high humidity, availability of hose, that is self-sown rices, stubbles, weeds, high nitrogen fertilizer application, low K application, incidence observed in fields which lack proper nutrient management. That is, for incidence, it is poorly managed fields, but for survival and development of the insect, we need a very healthy plant. And closer spacing favors Gall Midge incidence and development. And now with the growing of highly susceptible and high yielding cultivars, there is a flare up in all the command areas. Reports are there that high organic menus favor incidence. Type of planting has no influence. This is one statement from our cooperators from Nellore. And direct seeding has less incidence in Monkampur, Sambalpur, Chiplima, Gangavati, and Ambikapur. Though it is not nil, it is less incidence. Late planting was reported to cause high incidence as already established, but except at Chiplima, where early planting is causing high incidence. But the factor is, if you look at the farmer's point of view, farmer cannot identify the infestation until a gall is formed. He will identify once the gall is formed and or at the time of weeding, and he will not be able to differentiate an active gall or an insect emerged gall or a parasitized gall. That is the situation. So by the time he realizes it, one generation would have been completed. And this is a pupil case where the gall will be there in the field, but it is an already emerged gall. This situation is very important to judge because the timing of chemical control is timed based on the presence of insect in the field and what is the status of insect in the gall. Now, summarizing the peak incidence of Gall Midge, if from all the areas we had 18 locations, I have summarized for 16 locations. We can see that it starts slightly from July, where we notice it, but peak incidence from August to October it will be there. And in most of the locations, it is August and September incidence. After that, the incidence will be there, but the damage will not, we will not recognize the damage. And again, January, February, and in some locations up to March and April. And most of the people, they have reported that up to 30 to 35% silver shoots in susceptible varieties, causing a yield loss of 25 to 30% yield loss. Now, coming to the survival strategies, very many weeds have been reported depending on the cultivation systems that are prevalent in their locations. But if I look at the literature that is already available, grasses are they serve as alternate hosts. And through our confirmed host plant host transfer studies, we have confirmed that Echinoclova, Crusgally, and Plasmodium, they serve as alternate hosts. And wild races do have galls, but we don't know which species is infecting these wild race species because we have not conducted the host transfer studies. There are many grasses with galls which are prevalent, and there is a increase in incidence in these grasses and in the rice also simultaneously. And during host transfer studies, there is a variation in the symptoms of damage where you can see panicle gall forming, and when you infect, when the appropriate stage is not available. Each spikelet is also turning into a gall on echinoclova. But with biotype 3, this was a study way back in 2004. So it will not develop, but it will give a HR plus reaction. But we are not sure of the alternate host status of dicots which that are prevailing in the rice ecosystem across all the locations. If we look at the survival in stubbles, with machine harvesting, the stubbles are very long. They are able to survive within the stubbles, and when the sprouts are there, it is stimulated and we can see galls emerging from the leftover stubbles in the field. This would be a source of inoculum for the next generation. And once the nurseries are ready, the insect would go and infect in all the endemic areas. Now, with regard to the insecticides, we can see that 3 insecticides are very commonly used, and these are very effective because they have a fumigant action. And in Chiplima, they say that Azadiracta, Indica, and Chrysanthus colinus leaves also are helpful in this control. But there are many insecticides which the farmers use, which are very ineffective except for vinolphos at some point of time. And these are the farmers use based on the recommendations from the dealers. And here there is one more, that is chlorpyrifos granules are there. Previously, 4-8-10G was very effective. This was phased out. Now the farmer is familiar with the 10G. So he doesn't know whether it is 4-8 or chlorpyrifos. So instead of 4-8-10G, chlorpyrifos 10G is given, but this is not very effective against Gall Midge. So most of the farmers are using this 10G, which is causing less control of the insect. Now, if you look at the IPM strategies, these are the various options that are available. That is optimum sowing time, then pest monitoring, field sanitation, removing the weeds, N management, nitrogen management is very important, and optimum spacing, as I've already spoken. Natural biocontrol. Here in this case, we have Platycastor oryzae, which increases in its population in the late season. So in the early season, it is very low. So the appropriate insecticides that we use would conserve or help in perpetuation of this important parasitoid. And need-based insecticide use, I have already discussed. Only available option is carbofuran 3G or fipronil at our disposal, and the farmers have to be sensitized on this particular aspect. Cartap hydrochloride granules are also effective. So the final option is we have to look for the durable host plant resistance, so that at least it will lower the damage to some extent. And we have a very good screening protocol where we collect from the endemic areas insect and we rear on the susceptible host plant, that is TN1, and simultaneously we release it into the screening trace. So, and here what is important is after the release of the insect, it has to be subjected to the humidification so that the maggots that hatch will go into the plant. And we have to do both raising of the plants and screening simultaneously, which is very labor intensive, and it is highly sensitive to temperature and humidity requirements. And once we lose the culture because of low temperatures in the late season, we again have to wait till the next July or August for field inspections to collect from the field. We can also do field screening by releasing the adults in the field with and this laboratory screening. We have 25 females with 10 males standardized by Vijayalakshmi. And for maintenance, for each tray, if we put 3 to 4 mated females, that would give 9 times the amount of culture which we have released. This is the standardized protocol from IIRR where we go. And if you look at the interaction, what is happening, this is the HR minus interaction where the maggots are dead as soon as they enter. And we can see many maggots enter and they die, but only one maggot survives in one tiller, and they move from the growing point to different tillers. This is in W1263 and Gravia, both GM1 genes. And in the successful interaction, you can see that galls are formed and the adult emerges. This is a HR plus reaction with Abia. But there are other interactions which we have been observing. Sometimes in the gall trays, the insect, the gall development will be there, but the plant also will not be able to survive because it will not be able to sustain the attack. We cannot either grade it as susceptible also because there will not be any gall chamber formation and the plant dies. And in some cases, the gall formation will be there, but the insect will not be able to complete its life cycle. The pupae are dead within the gall, as you can see here. This situation we are seeing in some of the gene differentials at Jagatyal for W1263, where if you dissect out the galls, gall tubular formation will be there, but the insect is dead within. So there is a change from HR minus to insect is able to go develop till, but it is a chronic antibiosis. And there is delayed susceptibility. That is, it will escape at the vegetative say 30 DAT, but slowly there will be gall formation, or sometimes it will dry. So both situations are available. And some of the resistant lines in the reproductive stage, they put forth galls. That means in the vegetative stage, they offer resistance, but in the reproductive phase, they are able to put forth galls and the insect is able to survive. So this is the various situations we are facing with respect to resistance. Now we have the donors available, though it is single gene. Right now there are 12 genes, and till date we thought that GM1, GM3, 4, 8, and 11 are effective. But right now, if you look at the data, GM1 and GM8 are more effective. And we have 3 genes that have been cloned, and already 7 biotypes based on their reaction to different host plant resistant differentials. 7 biotypes have been characterized, and with the various data, Dr. Bentur and Nair have come out that CM4 with GM8 is the best combination for the pyramiding of the genes. This is a HR plus reaction and this is HR minus reaction. And for the deployment, we have lines in the background of MTU 1010, improved Sama Masuri, Akshay Dan, WGL 14, Tellamsa, Swarna, and Naveen with these 2 gene pyramided lines. And some biomarkers are also available from metabolomic studies with respect to Rice Gall Midge interaction. And for the past in ICRIP, we have been phenotyping extensively, and I will only just give a glimpse of it, how much material we screen across locations and in our IIRR glasses. For nearly a decade, nearly 11,000 and more lines in NSN1 and NSN2 we have seen. But since it's not a targeted breeding program, we have less than 0.15% lines which are resistant. That too for 2 or 3 populations only, 2 to 4 populations, but not all the populations tested. And we have some good donors which are conferring resistance across 6 to 7 populations. And we have one BPT mutant and sources from Oryza glaberrima also now, and some of the pyramided lines which are holding true. But if you look at the marker point of view, some of the lines, though they are marker positive, they are not conferring resistance across the populations. Only 1 or 2 lines are good. And sometimes marker positive points, they may be marker positive, but phenotypically susceptible. And that is the same thing. Marker positive, but susceptible at reproductive phase. This also we are seeing. Presence of many genes is influencing the phenotype. Like when we are pyramiding many genes, whether the final outcome is HR plus or HR minus, that is also making a difference. Mostly, already our seniors have reported that HR plus is epistatic over HR minus. So right now, if it is HR plus, whether it will hold for many years or not, I'm not very sure. But this situation we are facing. So Dr. Bentu sir was asking about the primers. Have you amplified some of the gene primers? And here we can see this is a glaberrima line. GM1 R alleles are there, but here if you look at the phenotype, it is not conferring to all the populations as it should have been. So there is some variation in the expression of these, though it is primer positive. And now coming to the insect part of the story, the maggot, it feeds on the growing tissue. In a vegetative phase, it is a pest, but some reports are there that if the insect is not able to find a suitable stage of the crop, it can also infest panicles, as this has already been reported by Rajaman and Pasalu way back in 1969, that is panicle galls also can be formed if the conditions are highly favorable. And we have the knowledge on the transcriptomics of the salivary gland of the Gall Midge. Then mitochondrial genome, we have 5 biotypes have been characterized and we have markers for that. And Gall Midge microbiome has also been studied. Wolbachia in the adult and pupa Wolbachia is associated with adults and pupa, and Pseudomonas in the larval stage. And the draft genome is available but still unpublished. Now, coming to the monitoring of the Gall Midge populations, this has been a continuous activity at IIRR formerly and IARR at present since 2 decades, more than 2 decades. Previously, as already told, 7 biotypes have been identified based on the reaction to a set of host plant differentials. And now we have increased it, the number has been increased, but and we have also included 3 lines with GM1, GM3 and GM8 in the background of ISM for monitoring, so that if the same background is there, it would be easy. And across years, when I was compiling, I could see that only INRC 3021 and Agani are holding good in terms of across locations, and we can see that Kavya and W1263 are better. So GM1 and GM8 are holding good. And other differentials, there is a variation in the performance of the other differentials. Some few years ago, we were thinking, because this data, what we were monitoring is at the research stations where large amount of research material and breeding programs are directed towards Gall Midge resistance. So this one set of differentials we have evaluated at Warangal in the farmers field. And I could see that this experiment has been going on for 4 years. And we could see, and I'm showing you 1 year data here. We can see here that there is a less virulence when it is in the farmer's field. This field is 30 kilometers away from Warangal research station. And you can see MR1523, there is a difference in the reaction. RP, that is with GM3, there is a difference in the reaction. Even the GM8, there is a difference in reaction. And in the farmer's field, it is still, it is holding true. And this is a new donor which is holding good. For the past 4 years, it is consistently performing and it appears to be having GM8 gene. And these are the locations currently which are where these genes are promising. Like Kavya is promising only at 4 locations. It is surprising to note that at Gangavati, ARC 6605 is better, and at Katak, ARC 5984 is better compared to many other donors. And this is the virulence monitoring where single female progeny test is done. Previously, we used to do with RP, GM3, GM8, GM1 and purple. Since 3 years, we have put to see how the pyramided lines would perform. And this is a setup which I'm showing you with one of our cooperators, Dr. Shravan from Warangal, who does each cup where the differentials are shown like this, single female will be released and evaluated for the damage. And this is done across minimum 6 locations, 4 to 6 locations based on the interest of our cooperators. And we can see we have introduced the Akshadan since 2 years. And if you look at the scenario here, we can see that Jagatyal W1263 is still holding, Agani with no virulence. But if you when RP is grown, the damage is very high. But with Akshadan, you can see at Jagatyal it is not virulent, but at Gangavati, then at Patambi, Ragolu, it is highly virulent. So again, deployment of these genes should be done very cautiously. That is my opinion. So this is a 1 point for discussion. Markers are available for all the genes, and we can go ahead with the available genes and the markers to elucidate the mechanisms. And functional markers are available for all these 3 genes. They have been cloned. So if you look at the pathways, it is mostly NBLLR and NBRC gene. I think the Gawans who have elucidated these paths are sitting there for the, and we look forward to their comments. And von Willebrand factor in GM1, it is getting upregulated in different lines that we have tested, and it is still consistent with GM1 gene. And in RP, where the HR plus is there, lipid peroxidation were upregulated, and the marker metabolite, that is azelaic acid, was accumulated during HR. So this coincided with the accumulation of the neurotransmitter, that is gamma aminobutyric acid, leading to the death of the insect. This mechanism has elucidated in GM3 in RP2068. Now coming to the varietal profile, this is the thing which is available in our database. I've picked few of the things to just bring it to your notice, and this we have got it from our cooperators from the questionnaire, and we can see that available resistant sources are there, but farmers most often they use high yielding fine grain quality susceptible varieties. That is one thing. Still in Kerala, Uma and Jyoti are cultivated. The ones which are marked in red are the resistant varieties that are under cultivation. MO4 is in coastal Karnataka. Previously, Brahmawar used to have large incidence of Gall Midge. Now they say that the incidence has very much disappeared. Insect is there. See, when we go to light trap and we collect and we do a single progeny test for virulence monitoring, we do get few insects, but it is not very much high to cause infestation on the resistant varieties. In Telangana, it is Pranahita and Kenin. And in here in Chhattisgarh, Mahamaya, Karma Mashuri and Samleshwara, all these varieties are there. We have tested Mahamaya and Karma Mashuri out of my interest for the past 2 years. I feel Karma Mashuri is gaining susceptibility, but Mahamaya is still holding. So Karma Mashuri may back down. I think it has GM4 gene. In Titaba, it's not very alarming, though insect is there. Tamil Nadu, yes, these varieties are there, but people use private varieties for various reasons. Sakoli, we have very many tolerant lines for Gall Midge, which are under cultivation. Sakoli 9, MT10 is susceptible. I don't know why they have given. They say it has some level of tolerance. SYE 2001 and PDKV Kisan. Now, we had circulated a question. If you are given an option to introgress some of the lines, what are the lines which you would like to introgress? In which background you would like to introgress? These are the lines suggested from Tamil Nadu, from Telangana, and from Nellore, and from Sakoli. And they have also given details of the specific grain types, what type of grain they need, then all the other parameters, which I have not put it here. But and they have given all the details on what type of duration they want, what grain type they want, what is the amylase content they want. They are very clear on what background they have to choose for the introgression of the lines. Now, coming to the biocontrol, the farmers, they cannot differentiate a parasitous gall or an healthy gall or a parasites, because here I want to share one of my experiences, because now with the digital media that is coming, people just send the photographs and they say, you tell the control measures. So in that context, from Nellore I was conversing, I said, you cut open the gall and see if the insect is there, then you can go for insecticide application. But already they had applied chlorpyrifos and they sent me the photographs. And once they cut open the gall, I could see that most of the natural enemies, they were dead. They only know that insect is there. They don't know whether it is a parasite, parasitoid. They were predators and both parasites which are dead. I was very much astonished how we talk very loosely and how much difference it makes in the decision making. So cautious dissemination of knowledge is very much essential. And here, the one aspect is sex pheromones. Yes, female, here in this institute, Dr. Brentwood has demonstrated that female Gall midges they attract males and it can be utilized. Afterwards, Hall has reported that this structure of 2,6-diacetoxy-heptane is available and this can be utilized, but no further work is available. So this point can be explored for successful management of this pest. And the way forward is, with all this understanding and the knowledge available, we need to understand the behavior of the pest, sharing and evaluation of the land races and wild access. Just because the 12 differentials are not becoming not up to the mark, it doesn't mean that new biotype has come. We need to really evaluate more land races and wild race accessions to find more genes if they are conferring any resistance. Then sensitizing the farmers in the endemic areas to go in for granular application at the right time, because unless it has a fumigant action, it cannot kill the adults or the pupa that is traveling up the gall. And to differentiate an active gall from an emerged or parasitized gall, to identify the parasites and predators which build up late in the season. Protocols to be developed for precision phenotyping using 3-cap panel to identify lines with the promising genes and the haplotypes, to dissect the mechanism of tolerance for appropriate breeding strategies, then studying the effect of background on expression of resistance and tolerance, the compatibility of a specific type of plant resistance with other pests. Here, Gall Midge incidence, it will favor for stem rot. And there is also a report from our cooperatives that stem borer there is a displacement of stem borer once Gall Midge incidence is there. But BPH incidence suppresses gall formation in the early stages. These three are the things which have come out from the discussions. And location-specific deployment of resistant varieties with good yield and quality traits has been obtained. And monitoring of the studies on Gall Midge virulence to continue, and exploring the possibility of using sex pheromones for Gall Midge management, and also decision support system for Gall Midge prediction, infestation prediction. Thank one and all for your patient listening. Director ICAR IIRR for giving me this opportunity, PA of the Entomology program, expert panelists, fellow colleagues and cooperators, and to all the scientists in the research centers who have shared their knowledge to the questionnaire and my scientific practice.
And also its resistance aspects. Dr. J.S. Bentur to kindly give us a brief observation or remarks for the next 10 or 15 minutes. Sir, do you have a presentation or it will be through a talk, sir?
I have a small notes which I'll share on the screen.
Okay, sir. Okay, sir. That'll be fine.
I have not made a PowerPoint, but I have just a few scribbled few points.
You can go ahead, sir. I can start sharing.
Yeah.
Thank you, sir.
Good morning, everybody. First of all, am I audible to you?
Yes, sir.
Yes, sir.
Yes?
Sir, you're audible.
Thank you. First, let me thank the Director Sundaram and Dr. A.P. Padmakumari for involving me in this brainstorming session. I feel it is a very timely activity, and I wish that there will be more to follow it. It's good that we begin with the Gall Midge. And I also would thank and also wish my colleagues, Suresh Nair, my collaborator, and Dr. Arvind Kumar, and Dr. Durga Rani Madam. So what I would just put some thoughts in a write-up. Can you see my screen? I'm sharing my screen. Can you see that?
No, sir. We are unable to.
I'm telling it share. Allow Zoom to share screen, open system. No?
No, sir.
No, not yet.
Okay, then forget about it. At least I can see, and I will follow it up. You will see my voice. So Gall Midge is a typical buck boost pest. Sometimes the incidence will be so high there will be outbreaks. At least in my experience, we had an outbreak in 1975, a report of which I read. Dr. Kalode visited the Sakoli area. That was the first one. Then in 1985, I was also part of the team. There was an outbreak in Srikakulam area. Then in 1995, there was an outbreak in Monkampo. I'm roughly putting these dates, not exactly the precise year, but every 10 years. Then in 2005, we had an outbreak of the new biotype in case of the Warangal. And probably I'm not keeping a track after that. Maybe there was something. And now again, you have a situation, alarming situation. But what I would assume that Gall Midge is a typically manageable pest, though once in a while we get certain hiccups. And I would call this situation as a slightly hiccup. And as summarized by Padmakumari, we have several options. To begin with the cultural management, where early planting can escape the incidence of the pest, though it is effective, but it is not practical. The second approach of chemical control, which she has identified a set of chemicals, though they are effective, but not advisable, and the cost-wise, they are not suitable. So that comes to the only choice available is a genetic approach, development of resistant varieties, identification of new sources, new genes, developing and tagging and mapping of the genes, developing markers, using markers for pyramiding the lines. All the progress that has been made in the past 2 to 3 decades, I can confidently say that research is one step ahead of the Gall Midge in evolution of virulence. So though it is a satisfactory fact, but we cannot be complacent. So the research have to be geared up to face further challenges. Then what is the situation that alarmed now to have this brainstorming session? So I asked Padma to send me the latest reports to see whether there is anything that is unusual or abnormal in the recent developments. And the reports of 2022, I just rapidly went through, and then there are certain information I'm recapping, though Padma has already covered that one. In case of Gall Midge screening trials, you have certain lines which are good at 9 out of the 5 to 6 tests out of the 9 tested here. When you look at these promising lines, let me see whether I can scroll my own slides here. Yeah. The best entries here reported are IBT WGL3 from Durgarani Madam. It has done well in 6 out of 12. PTB21 is a landrace from Patambi. It has also done well. And then WGL 119, which has also done for 6 out of 12. Though I'm not aware about the gene combination in these both WGL and IBT WGL, but it says that gene pyramids are working. So we need to know more new combinations. I was told that there are many in the pipeline which are being screened. So this will be our immediate rescue point. So some of these will be able to help us to overcome the present hiccup. Then in Gall Midge biotype monitoring trial of last year, we have several gene differentials and combination of genes. In there, we have 3 donors like Agani, which has done good in 11 out of 12. Same thing is INRC 3021 and also INRC 17470, 9 out of 12. All these are carrying GM8 genes as confirmed. But then just now Padma told that even the new landrace INRC 15888 also carries GM8. So also the RP 5925-24, which has done good in 7 out of 12, this also carries GM8. So the take-home message at this stage is that our best bet is GM8 right now. And in addition, as Padma was telling that, this can be used in combination with GM4 or GM1. So the options are still good, and then the markers are there. breeding with the pyramiding with marker-assisted selection is the intermediate solution for this problem. Then coming to the studies on virulence composition of the Gall Midge populations. Again, last year's data, I wish I could have shared the screen, but it tells me that there are two separate the populations. One in Kerala, for example, Monkampo and Patambi, there is a high percentage of insect virulence in case of the GM8 and GM4 and 8 combinations. So this combination does not do well in Monkampo. So let us take that fact for granted and look for the other combinations in Kerala area. But in case of AP and Telangana, the results of Ragolu and Jagatiyal and Warangal, it shows that, just a minute for me. GM1 is still holding good there. The GM, though there is 20 to 30% of the population is showing virulence, this still can be managed. So we can still use GM1, and GM8 is very effective. A combination of GM1 and 8 or combination of GM1 plus 4 plus 8 will be mostly good for Karnataka, Andhra Pradesh, Telangana, maybe part of Tamil Nadu also. So what is needed here is extensively test these lines, not necessarily in the large-scale farmers trial, but in the coordinated trials, and those which are the varieties which have carrying these genes in combinations. So this could also be an intermediate solution for the next coming few years. Then coming to the gene markers, the molecular tools available, Padma has already summed up the availability of either gene-specific or gene-linked markers for all the 12 genes. And maybe some of the genes may not be very precise. What she was telling is marker positive, but it is susceptible. So there could have been a recombinant forms there. And also in combination form, some of the genes like GM4 and GM8, I see that GM4 may not be effective in combination in some locations. We need to study this, but let us not assume that the gene combinations in general are not effective. They are really effective, and new combinations need to be generated. We have not used GM3 much in our breeding program, which is also a good gene. We can try for that. And then coming to the gene-specific markers, I have one suggestion to both our director Sundaram and also the Padmakumari and the team, that in addition to the phenotyping information for the lines, at least the promising lines in GMS, GMBT, we need molecular phenotypes for phenotypic data also. When you say that some plants are showing damage, I would like to know whether these plants did carry the gene or they are mixtures. So this data can be generated. It's not very expensive or not very time consuming. Only thing is we need to make up our mind to generate this. So that gives us a better understanding and better interpretation. Why a gene did not perform well or why a gene combination did perform well. Then coming to the plant candidate genes. Now the next generation or the long-term solutions or the planning should be for genome editing. It is much easier now, much realized, though it looks like a fancy research today or more academic and more publication oriented. I'm sure in 5 years down the line, we have products of these gene edited. If we have to edit genes for Gall Midge resistance, first, my focus would be on GM3 and GM12, which are recessive genes. In general, we consider that recessive genes are the non-functional susceptibility genes in general, but it may not be always true. So let us first attempt, at least on a trial basis, by silencing or editing these two genes and see whether it will confer the resistance. Then there are susceptibility genes which have been reported, and Padma mentioned about von Willebrand factor protein, Rawat et al. Though it is active in the incompatible interaction, silencing would suppress the incompatibility and induce the factors. Like that, one more gene she has identified, Nidhi, that is transcriptionally controlled tumor protein. This is the one that leads to the cell proliferation. The maggot feeding leads to the nutrition tissue formation in the gall and ultimately gall chamber formation. So in addition to that, there are several genes. As we often tell that susceptibility against Gall Midge and the rice is not a default phenomenon. You need the cooperation or active participation of several plant genes for insect to survive. So any one of these, the team of genes or suite of genes, if they do not function, the plant cannot support the development. That means that it becomes resistant. So whatever genes we are focusing, they are all defense genes. We are not focusing on the genes that help in formation of the tumors or the galls. So these are our likely targets for the genome editing. And then there are also the insect genes that can also be targeted either by editing and then a big question, how do these gene edited insects will prevail in the nature? We can also do the gene silencing through expression of RNA, double-stranded RNA in the plants. And these, there are two candidates. Suresh will be able to speak more about that. These are effector genes or salivary gland secretory proteins like oligosaccharyl transferase, which is induced during the compatible interaction sevenfold, and also NDPK, nucleoside diphosphate kinase. So these two genes are the likely candidate genes which can be either edited in the insect or they can be silenced through the plant mode. So this should be our long-term goal. And there is one more thing that we can think of is CRISPR-Cas9 mediated gene drive. This is a powerful technology that is coming up. What I would like to suggest, Dr. Sundaram, is that this is the time that we need to develop expertise in these areas of research to face not only the problem of Gall Midge, but other pests as well. So I have highlighted immediate, intermediate, and long-term solutions or strategies to manage rice Gall Midge. Thank you very much. If I have any questions now or later, I will take it. Thank you so much.
So what I suggest is at the end of the panel discussion, we can have a question and answer session. But in the meanwhile, I invite all the participants to, if you have any query, you can put it in the chat box so that it can be read one after the other and then answers can be solicited from the experts. Is it all right, Padma Madam? Okay. So I now invite Dr. Suresh Nair, Arturo Falaschi Emeritus Scientist at ICGB. Sir, you can go ahead and then give a present. So you have any slides to share, or it will be an extempore, sir?
I have prepared a slide, and
Okay.
I find that Dr. Bentur and Dr. Padmakumari has covered our work so well that I probably don't need to show the slides. But nonetheless, I'll quickly go through them.
Okay, sir.
But I'll spend some time on the last few slides because that talks about the way forward.
Okay, that's fine, sir.
Is that fine?
Yeah, that's fine, sir. You can go ahead and then share your presentation, sir.
Yeah, just a minute. Are the slides visible now?
Yes, sir.
Go to full screen, sir. That's the only thing.
I thought
Yes, we can see your
full
slides.
screen. Yeah, okay.
Yeah. Now you're able to see, sir. Full screen. You can go ahead, sir.
Yeah. Yeah. So go back to my slides. Dr. Padmakumari already talked about the work that we had done earlier. At ICGP, we have mapped and tagged 4 different Gall Midge resistant genes and show their chromosome location. And the quick question that we were asking subsequently is we were looking at secreted salivary gland protein that Dr. Bentu just mentioned that we had done some work on that. And why secreted salivary? Because if you look at the interaction between rice gall midge and the rice plant, the entire interaction happens in the larval stage. So obviously there is something being secreted by the Gall Midge larvae that the plant is able to recognize. So that is one of the reasons why we looked at SSGPs. Here we looked at RP2068 and we isolated one of the different SSGPs available, that is defense against cell death, DAD1 gene. And we show that if you take DAD1 from the rice gall midge larvae and clone it, and you get the protein expressed through E. coli system, and you inject it into plants, you can get a hypersensitive response. And we looked at another protein which is NDPK. We find that this particular salivary gland protein probably increases the cell dimensions. So that is one of the things that the rice plant, gall midge requires of the rice plant to do for it, basically to work for it and produce the gall. All this work is published, so I'm not going to spend a lot of time on this. Now, one of the things that
Sir, we are unable to hear you. Hello? What's the problem?
We can't hear you, Dr. Nair.
Sir, we lost your screen also. We are no more able to see the PowerPoint slides presentation.
Okay.
Can you see me now?
Yeah. Yes, sir.
Wonderful. Yeah. Sorry for the interruption. Yeah. So what I thought was the way forward, I look at 2 things. One is the culture and the other is the molecular biology of the insects. And before I go on to these 2 major topics, I'd like to highlight one thing that Dr. Padmakumari mentioned in her summary, that resistance genes are breaking down. At this point in time, what I would have liked to know is there any change in the Gall Midge population structure? And this can only be monitored using the different markers that we have, the different biotypes of Gall. So these screenings for deviation in the population structure is very important to reassess the pyramiding strategy that we have done before we say that a particular gene pyramid line has broken down. While it is good to rely on field-based screening, it is equally important for screening rice lines under greenhouse-raised cultures. The reason for this, I need not re-emphasize, I'm sure it's very clear. And more emphasis for field-based collection of flies from the field on an annual or biennial basis. Now, are the same population causing the damage, or more than one type of population is present? Now, markers for these biotypes are available, And I think we should make use on a routine basis to find out whether there is a change in the population structure on an annual or a biennial basis. During the course of our work, a collaborative work that we did with Dr. Bentur and Dr. Arvind Kumar and Dr. Sahu at the National Rice Research Institute, we found that it was very difficult to get Gall Midge cultures. Brown plant hopper was never a problem, but Gall Midge cultures was always an issue. So we need to have a glasshouse cultures of the different recognized biotypes of Gall Midge all year around. I know it's a difficult task, but this needs to be done now. Without the screening for resistance, it becomes a major problem. Regional station could be asked to have their own culture, whatever is present in their particular locality, so that whenever a culture dies, it's a very normal and routine process I realized over the years, that we need to have these populations back. It's very easy to get the population from these regional centers. And as soon as any deviations from these patterns are available, it should be noted and told to all the people concerned. Coming back to gene combinations. Now, before we declare that a gene combination is no longer valid, as I previously mentioned, we need to know the population structure. Now, another point that Dr. Bentur alluded to was looking at CRISPR-Cas method of gene editing of GM3 and GM2 to susceptible lines, to susceptible genes. Now, in the Rice-Gall Midge rice interaction, we still do not know what the so-called susceptible gene does. So we need to clone these genes. Unfortunately, we have only very close markers or genes very adjacent to this particular genes, and then we need to find out and characterize these GM3 and GM2L before we embark on this editing process. With these few words, I think I'll stop. I'll let the other panelists talk about what they have to say. And we can take more questions as we go. I'm sure there'll be more discussing points that we'll come across. But these are the points which came to my mind immediately, which I thought I'll share with all the other people. And I once again thank Director in the Indian Institute of Rice Research, Dr. Padmakumari, and all the others who had me on this panel. Thank you.
Thank you very much, Dr. Suresh. I now invite, we can take the questions at a later stage, and I also request all the participants online and offline to kindly type your questions in the chat box so that they can be read out and they can be documented also. I now invite Dr. Shyam Kumar, Associate Director of Research, ZARS in Devahi, Maharashtra, for his brief remarks. Dr. Suresh.
Good morning, everyone. So respected Dr. Sundaram, Director IIRR, Dr. Padmakumari, for giving me this opportunity, and also my namaskarams to Dr. Bentur and Dr. Suresh Nayak and other panelists, so Dr. Arvind Kumar and others. So good morning. Actually, the great work has been carried out by Dr. Suresh Nayak, Dr. Bentur, and other entomologists, molecular entomologists like AP Padmakumari. And here I want to tell that we have used those that information for introgressing those genes by using those markers into our lines to bring the durable resistance. This is mostly the application part, and I'm also going to tell something about the Telangana state rice cultivation and how this Gall Midge is going to be changing from time to time. So that is the thing. Can I use the mouse? And here, I would like to tell about the contribution from the Warangal station. So because Warangal is the center, earlier it was under Angro, Acharya NG Ranga Agriculture University. Now it is under Professor Jaishankar Telangana State Agriculture University. So which has started the research on the rice Gall Midge, and which is a prevalent pest in irrigated, rainfed and also wetland environment, and particularly as earlier speakers mentioned that it is a early stage pest so it occurs mostly in the tillering stage, but now we observe some deviation also. So here, this was the first station initiated and named as the Rice Gall research, initiated during the year 1959-60. And which is also a national center for the breeding Gall Midge resistant varieties under the ICAR system in the year 1969. And from this particular research station, IIRR Warangal, the first Gall Midge resistant variety Kakatiya was released in the year 1974. And this was the station where this Gall Midge biotype deviation or the variation was observed, and the 4M biotype was reported for the first time for the new gene conferring resistance, this one. So, and also so much research has been carried out and related to the inheritance of the Gall Midge resistance, and several reports they have mentioned that it is a single dominant gene. As we know that there are among the 12 genes, 2 genes are recessive and the remaining all dominant resistance genes. And here the subsequent studies also, they have well characterized the biotype studies, and so many Gall Midge resistant donors were reported, and they have developed the IPM modules and developed Gall Midge resistant varieties against the Gall Midge biotype 1 at that particular point of time. And coming to the status of the area production productivity of rice, why I have taken this slide is, so after bifurcation, Telangana's state formation happened in the year 2014. So from that time onwards, if you look into this area, the area has been increased tremendously because of various things like the irrigation infrastructure facilities improvement was there, and the free power to the farmers, and the Raitu Bandhu and the Raitu Bima schemes, along with the access to the inputs and market and credit facilities. So here, the 142% increase in the grass value added by the agriculture and allied sectors were noticed in Telangana state. And these are the 2 important districts contributing towards the 8.4% and 8.3% of the total production of cereals contributed by these 2 states. And also this is the principal source. This particular sector is giving employment for around 55% of the population, and the contribution to Telangana's gross state value added. So this figure has been increased from 16.3% to 20.5%. So during this particular period, 2015 to 2020-21 statistics as per the Telangana State Statistical Abstracts. So this all happened because of the contributions from the rice breeders from various agriculture research stations and also several RARS. And here I'm proud to say that 26 varieties were developed during this period from 2015 to 2022. And there are several releases through the SVRC and also CVRC. And so here one point is with reference to the Gall Midge, I would like to tell that, so among these varieties, so many varieties are there, so many varieties are high yielding varieties, but they are having the resistance to other pests and diseases. But coming to this Gall Midge resistance, WGL 347 and WGL 1119, these are the 2 varieties having the Gall Midge resistance. The other varieties, earlier released varieties like Sureka, Siddhi, Bhadrakali from the Warangal, which are offering resistance against the Gall Midge biotype 1, they are in the seed chain to some extent. And there are several varieties released earlier by Warangal Center, which are also showing offering resistance against the Gall Midge biotype 1 at that particular point of time, which are not in the seed chain. Okay. So these are the varieties released from rice varieties released from Warangal. So why I'm giving this one is like, so the first variety is Kakatiya, followed by so many varieties were developed, and all these varieties are towards having the Gall Midge 1. When you look back, actually at that particular point of time, there is no way of looking into that particular gene. But now if you look into those, what is the source of the resistant gene? that is GM1, and mostly towards the GMB1. So, but we know that the Gall Midge 4M biotype is more prevalent in addition to this Gall Midge biotype 1 also from in Warangal, and also there is a GMB3 also reported at Warangal. So now these are the latest varieties where the GM4 has been introgressed into 347 and 1119. So this 1119 is the first mass variety which is going to. Earlier we have another variety which has been released in collaboration with the ICAR IIRR for low P. But coming to this Gall Midge resistance, this is the first variety developed through marker assisted breeding. So where the GM4 along with XA13 and XA21 were also there in this particular background. So this is going to be the proposals, release proposals are going to be submitted shortly. And here we observe that as the rice growing area is increasing and the production is increasing, but at the same time, so we are also worrying about the increasing incidence of this Gall Midge. So what could be the reasons for the change scenario? That is the changes in the climate because of the continuous rainfall and consequent congenial microclimate because of the increased irrigation potential and the spread of the highly susceptible varieties means against the Gall Midge. So though they are having the resistance for other pests and diseases, but they are susceptible to the Gall Midge. So that as other speakers also mentioned that the high tillering varieties are prone to Gall Midge. And the intense management of management practices, as earlier Dr. Padmakumari mentioned that the poor management attracts the pests, but this intense management practices retains the pest for the longer periods. The overlapping generations, this is another thing because of the availability of a host which is in susceptible tillering stage for longer periods. Because earlier when there is less irrigation potential or facilities, then the crop is in the limited area. Now there is sufficient water and the sowing window is not precise. It is extending so that the overlapping generations are going to be one of the important reasons for observing this pest even in the Rabi season. And another one is low parasitation. As we all observe that the chemical interventions are mostly followed by the farmers, and that too untimely chemical applications is going to result in loss of its natural enemy rather than the actual Gall Midge. So, and lack of farmers generally they don't apply the potassium as basal so that the plant resistance is going to be affected. And coming to some of the research related to this PZESAU with reference to Gall Midge, both at Institute of Biotechnology and also at RARS Warangal. So the Gall Midge resistance using the marker assisted breeding we have carried out, and for which we already know that among the 12 resistant genes reported, mostly in our studies we have used GM3, 4 and 8. We have targeted all the 3 genes, and for which in collaboration with Dr. Sundaram's group and also Dr. Padmakumari is our main collaborator. And here among the 7 distinct biotypes, Gall Midge biotypes, we have in Telangana, Gall Midge biotype 1 in Hyderabad, and the combined earlier we have is Andhra Pradesh also, because the varieties will spread both for the Telangana state and also Andhra Pradesh. So Maruteru is also having the Gall Midge biotype 1. And Jagitjala, Warangal, these regions, Gall Midge biotype is prevalent in Telangana state, and 4M is another one, Warangal. And in addition to this, Gall Midge biotype 4, which is from the Ragol, Srikakulam biotypes. So these are the biotypes mainly we concentrate 1, 3, 4 and 4M. So here the Gall Midge resistance is governed by gene for gene interaction, as we all know that, and no single gene provides resistance against all the biotypes, and no single biotype overcame all the resistant genes. With this, actually, we have started our work by taking the genes like GM3, 4, and 8, along with the markers which are very much the functional markers we have used in our studies. And so that by using these 3 genes which are going to offer this, we expected that they will offer the broad spectrum resistance against the biotypes GMB 1, 2, 3, 4, and 4M. So majority of the biotypes. So here, in addition to this, we also suspect some areas, some new biotypes also present based on the results we got, means the screening trials from Warangal particularly. We may expect some new biotypes, and some systematic studies are required in this particular aspect. And among the 3 chosen genes, the like GM3, 4 offers a HR plus mode, and also the GM8 offers HR minus mode. So this combination is going to work out well, so that we have chosen based on the discussion with Dr. Padmakumari, Dr. Bentu sir, we have started our journey. And this the material has been screened for Gall Midge biotype 1 at Rajendra Nagar continuously. And also similarly, IRRS Warangal, where this 4M biotype is very much predominant, which is very virulent. And based on our work, finally, we have these combinations of the lines, IBT-R lines, IBT-GM or IBT-WGL lines. So Dr. Bentur mentioned that IBT-WGL3 and WGL1119 have shown very good durable resistance based on the 2022 results. But before that, we have several IBT-GM lines also we have tested for 3 seasons, and they have shown durable resistance consecutively. And so these are the initial, this is the initial work through which we have developed certain lines where we have only GM4 gene as a part of network project. Along with Dr. Sundaram's group, we have developed this one. At that particular point of time, this GM4 line, means the lines which are having GM4 gene only, has shown resistance to 4M, but not continuously. It is not across seasons and across locations. Then we have attempted for adding subsequently GM8 and GM3 into this background, and there is subsequently another DBT project has come, and through which we have developed these lines. And based on these 2 products, we are not very much satisfied with the yield point of view. So resistance wise, now we are comfortable, but yield is going to be not, they are not out yielding the available high yielding varieties. So initially we have taken backcross method of breeding, but so based on the some of the limitations, we have switched over to the marker assisted pedigree method of breeding. So where when you maintain the large population, we can expect some transgressive segregants. With that, we have gone to this one. Even then, we are not very much convinced with the yield advantage. Then we went for the development of adding the yield genes like the grain number and the panicle length related genes. So finally, so now here 7 gene pyramided lines are there, and they are now almost they have entered into the yield evaluation trial now. So here, these are the various donors we have used. So now when Agani is offering very good resistance, but when we observe the recombinants, then we are not very much satisfied with the plant type and all. Similarly, RP2068 also. So that's why I can say that though we are not having a very good like crew genotype, which is going to have the high yield, but these lines can be very much used as a donors in the future breeding program. So instead of going for the Agani RT separately as the donors. So these are the markers which are they reported earlier, which are the functional markers we have used in our program. And coming to the resistant reaction of these rice lines, so we have tested our entries in the ECRIP system. And here a total of 13 lines at Rajendra Nagar. This is about the GMB1 result. And this is Odisha also and Chhattisgarh. These many lines, they have shown resistance consecutively for 2 seasons, 2 Kharif seasons. Similarly, those lines are also shown resistance against the Gall Midge biotype 3. And here, but there is some variation, like these have shown resistance at Jigichal, but at the same time, so those lines were not showing resistance at Ranchi. So maybe this has to be looked into. As per our results, the Ranchi might be having, means some new biotype might have emerged that has to be looked into. And coming to this, coming to the GMB4, like 38 lines showed resistance at Ragolu, and similarly 15 lines at Srikakulam shown resistance. And this is based on both the seasons results, only those I have given. And coming to this, when compared to the other numbers, this Warangal center, the number is low. 8 lines at Warangal shown resistance to biotype 4M under both the seasons. And again, this has to be examined further. There might be a mixture of biotypes or a new biotype. We have to look into the aspect. So cumulatively, by taking all the results, we have shortlisted to 3 lines showing Gall Midge resistance against Gall Midge biotype 1, 3, 4, 4M across locations, across seasons, and also they are also having the bacterial leaf blight resistance across. And here, so this already I said that Ranchi and Warangal, based on our study, we have to look into those biotypes. And this gene pyramiding proven to be a successful strategy for getting this durable resistance. So based on our study, which is MAB is a successful approach, and it is useful to track particularly the recessive genes. So even GM3 or GM12 also, because it is one of the advantages of the marker assisted breeding, the recessive genes can be tracked out. And here, these are the genes from our study. They are showing durable resistance against multiple Gall Midge biotypes. And as I mentioned, that large segregating populations study is recommended to observe the transgressive segregants. And here, in general, rice lines with multiple biotic stress resistance did not compromise in resistance against the target pests and diseases. But at the same time, we should not ignore that there may be some antagonistic effect of multiple genes on the expression of yield. So this could not be ruled out. So further study is to be detailed studies needed. Studies are needed to identify new biotypes, and also for them the new resistant genes are to be identified, and for them the markers are to be developed for breeder friendly markers are to be developed. So recommended to use these rice lines with multiple resistant genes as donors instead of using the original donors to have the superior recombinants in practice. So with this, I hope, so I will wind up. So thank you for giving me this opportunity. Thanks.